All right, so we're talking about the evolutionary consequences of sexual reproduction. And so let's look at just independent assortment. We're going to forget about crossing over. Synapsis and crossing over isn't happening on this slide. So we have dad in purple and mom in green. This particular organism has 3 chromosomes. The adult has 3 pairs of chromosomes. The gametes have 3 individual chromosomes. So dad makes a sperm cell, mom makes an egg cell. They unite and form a diploid offspring. These haploid gametes make a diploid, di meaning 2, a diploid child. Now it has 3 pairs of chromosomes or 6 chromosomes. So how could those chromosomes, now when it's time for this child to have their own kids, how could these chromosomes assort? How could they separate out to form their own egg or sperm, their own gametes? Well, I could get all dad, 2 dad, 1 mom, 2 dad, 1 mom in the middle, 2 dad, 1 mom on the left, etc., all the way to all mom. So with independent assortment and 3 chromosome pairs, there are 8 possibilities. 2 to the 3rd power. 2, meaning they are pairs, and 3rd power, there are 3 pairs. So it's the number of chromosomes of each one, the number of copies of each one, and for genetic simplicity, that's always 2. 3 is how many chromosome pairs are there? 2 to the 3. 2 times 2 is 4 times 2 is 8. So I've got 8 possible variations. All right. What about humans? We have 23 chromosome pairs. So if I did this exact same exercise with humans, it wouldn't be 2 to the 3 or 3rd power. It would be 2 to the 23rd power, or 8,388,608 possible kinds of gametes from every human being. When you make an egg cell or a sperm cell, you've got 8 million possibilities. That's just counting independent assortment. We haven't even included crossing over, which makes it 8 million to God knows what power. These numbers explode when you add crossing over. All right. Do I have it on the next slide? No. So what does that mean? Well, when my wife and I had kids, that means I had 8 million possibilities to contribute to our child. She had 8 million possibilities to contribute to our child. So our child had 8 million squared, or 8 million times 8 million possibilities. And eight million 388,608 times 8,388,608. So this is thousands, millions, billions, trillions. Just me and my wife had 70 trillion possibilities, not even counting crossing over, just with the chromosomal separation. 70 trillion possible kids. And it would be, in reality, it's way more than that. Way, way more than that. Orders of magnitude more than that. Because we haven't included crossing over. And that only includes the genes that me and my wife have. 70 trillion possible kids, and none of them are going to look like Shaquille O'Neal. They're all going to be short because we're both short. They're all going to look somewhere in the general concept of my wife and I and our families. So if you look at the human population with all of the genetic variation in the human population, the numbers are staggeringly big, incomprehensibly big, because one couple can have more possible children in terms of the variation that's there than all the humans that have ever lived on Earth, ever, by a lot. So that is, I think, a real magnifying glass on how important sexual reproduction is. The variation is mind boggling versus clonal reproduction, which makes a copy of itself and depends on mutation to make changes. Crossing over, we've already talked about that. That adds more of the variations. So the numbers I've just given you are minuscule, tiny compared to the reality. When you add crossing over, it's not 2 to the 23rd. I don't even know what it is, but there might be 2 to the 2300 power, because crossing over, instead of having only 2 chromosomes or 23 chromosomes, when you do crossing over, you might have hundreds and hundreds and hundreds, thousands possibly, of different variations. And then there's random fertilization. Oh, I did have the 70 trillion here. I forgot about that. Anyway, I've already explained this. I've already talked about the 70 trillion, not counting crossing over. So random fertilization means that the variation in sexually reproducing organisms, whether it's a rose bush or a broccoli plant or a human or a trout, it doesn't matter. They all have similar types of patterns. And again, go back to the idea of the wolf. When populations are being held relatively stably by environmental factors, wolves all look like wolves, right? There's some variation and you can tell them apart, just like humans can tell humans apart. They are all unique individuals, but they all look like wolves. But hidden within those wolves are tremendous variation that over evolutionary time or selection pressure, you can move those populations in huge directions. You can move wolves to chihuahuas. You can move some weed into broccoli. So genetic diversity is the raw material that fuels evolution and makes plant breeding possible. And no genetic process generates diversity more quickly than sexual reproduction. I'm going to take a quick break. Sorry about the interruption there. So I'm going to start sharing again. Anyway, no genetic process generates diversity more quickly than sexual reproduction. So we're looking at an example here. And this is the symbol for male, the symbol for female. And if I have a capital A, small a, these just are genetic representations of dominant genes and recessive genes. Dominant genes are a copy or a version of a gene that gives a recognizable trait, and the other copy doesn't give that same trait. So this could be, I don't know, anthracnose resistance, which is a bacterial disease. So this is anthracnose resistance gene allele. This is anthracnose susceptible. So if I cross a male, give the pollen to a female that has no resistance to anthracnose, and the male has one copy, what's going to happen? There's 2 possibilities for the male sperm. There's only really one possibility for the female egg. It's either got a small a or a small a. That's it. And so when I cross it, I'm going to get half anthracnose resistant children and half susceptible children. This is called a Punnett square. Punnett, Punnett square. and it's used to look at one or two genes. You get more than two, and it just gets too cumbersome, and there are better tools for analyzing genetic crossings. But this is a simple way of looking at what am I going to get. And you're looking at multiple genes if you're a plant breeder. So you might be doing Punnett squares one at a time based on what trait you're worried about at that moment. Or you might use, again, other tools. Here's another Punnett square, but in this case, it's a capital T, capital T, small t. So both the mom and the dad are both heterozygous. Hetero meaning different, zygous meaning their genetic makeup. Heterozygous. They've got a capital T, small t. They're different for their two copies of that particular gene. Different, what's called alleles. An allele is a version of a gene. Remember, mom and dad both have eye-colored genes, but mom had the green allele and dad had the brown allele. Same thing. So in this case, what happens? I can fill in the Punnett square, and I get 3/4, 75% of my children are going to be, let's say, I don't know, this is another disease resistance gene, just to keep it simple, or some disease that starts with the letter T. I'm going to get 3/4 of my children that are resistant, but only 1/4 is going to have what's called a genetically homozygous, homo the same, zygous genetic makeup. Homozygous resistance to this, and 1/4 will be homozygous susceptible. Again, you can use the Punnett square to look at what you're likely to get. A very good tool for simple breeding problems. And here you can see what happens, how it gets pretty ugly pretty quick. This is combining an R and a T gene, whatever R stands for and whatever T stands for, doesn't matter. But you can see how quickly it exploded with 2 genes, and with 3 genes, it's ugly. With 4 genes, it's completely unmanageable. Let's look at some genetic traits. There's dominance. Dominance means a gene or an allele, a version of a gene that is dominant over another version. So for example, in this particular chicken, the black chicken is dominant over the white chicken. So if I take a homozygous black chicken and cross it with a homozygous white chicken, you notice 2 copies of the black gene, 2 copies of the white gene, what am I going to get? Well, let's assume this is the male. The male can only donate the black gene. The female can only donate the white gene. And so the children are going to be half and half. Now, what if I take another cross between these? What am I going to get? I'm going to get 3/4 black, 1/4 white, but only one of the black is true breeding black. That all of its children, assuming it's crossed with another black one, all of its children would be black. It cannot donate a white gene because it or allele because it doesn't have it. And there's intermediary heredity or intermediate dominance, where you get something in between. Black and white make gray. Red and white makes pink. And so you can see what happens when you get the heterozygous form. You can see it. In the dominant recessive model, you can't see it. All you see is the dominant one. You can't see the recessive. It doesn't show up at all. All I see is black chickens here, even though the recessive or the hidden allele is there. I can't see it without doing genetic testing on a sample, a DNA sample from these chickens. All I see is black. But in these chickens, it's different. They have a different coloring model with an intermediate dominance or partial dominance. Intermediary heredity, where a black allele and a white allele makes a gray. And that happens in many cases as well. In some flowers, in some flowers, red crossed with white gives red. You can't see the white. You don't know. If I've got a red flower, is it true breeding red, or is it half red, half white? And you just can't see the white. And other red alleles, you get one red, one white, you get pink. And you can tell that it's heterozygous like this, where these are gray as opposed to black. Okay, that's dominance. But most traits aren't the simple black and white as we saw in the last one. Most traits are complicated. We call those additive traits. So things like your height, your weight, the shape of your skull, the length of your arm, the color of your skin, on and on and on. There's all kinds of variation, all kinds of range. It's not just this or that, black or white, red or pink or whatever. It's not so simple. And so it takes many genes, multigenic, in order to get the actual, what we call the phenotype, or the visible type of plant or human or fish, and see what it really looks like. Adaptive qualities are very important in additive traits, because the adaptability, all of these variation of traits are really important. Having lots of genes, lots of genetic material that affects height. It's not just you're either super tall or you're super short. There's everything in between. And that offers a huge amount of variation for the population to adapt to its environment. So, we've already talked about these selection traits earlier in this lecture, but, now we have to start making decisions and making choices, making measurements, scoring them, doing laboratory tests, doing field tests, and looking at all of these things. How does it work with pest disease? Does it have the disease resistance I want, the yield, the color, the uniformity, and all of those things that are important for me in either a plant breeding standpoint or as a grower. And that brings me to an important point. Why the hell did I do all of this? Why am I telling you this? Because as a grower, even if you don't care about plant breeding, it's beneficial to you. You make better decisions if you understand the business of the breeders and the seed companies that are trying to sell you something. You understand what's being developed, what's available today, what's on the horizon as a new variety that might be released next year or the year after. And by understanding their business and the genetics that go into breeding, you can do better trials on your own to test things that you might want to grow. And you might be able to be more a more knowledgeable consumer, to have better conversations with your seed dealers to get what you want. This is actually a trial I did in Texas with spinach, and you really can't see a whole lot of difference there at first glance. It just looks like a bunch of small plots with spinach with a whole bunch of stakes in front to mark them, label them, and I was doing an evaluation of a lot of the genetic material I inherited for this project. So I was growing it out and looking and scoring and getting some baseline data on what was the germplasm I had to start with for my breeding program, very early in my breeding program. There were some breeding lines in here, but it was mostly stuff just being evaluated that I inherited. But when you go up closer, you'll notice there's a lot of differences. These arrowhead shaped, pointy, light colored Asian style spinach leaves versus a very crinkly, what's called savoid leaves, dark forest green, very different. Those were in the same field in the previous picture, but you can't tell a lot of the differences until you get up close and you look and you measure and you get down in the dirt with them. Here's some variations in leaves. The one in the middle was a problem because it's too round and cup shaped. What happens round and cup shaped? You cut them, you have to rinse them in some sort of a wash cycle. in order to make a bag of baby leaf spinach. And then you put them in a dryer, which is a giant salad spinner, basically. And what happens when a cup-shaped round leaf is being spun in a dryer? The centrifugal force is going to force it to flatten out a little bit. And because it's round, it splits the leaves. Now I have a bunch of split leaves in the bag to start molding. And so one that had a nice shape, but a little bit less of a rounding, a little bit of a point to it, they flattened out much better inside the spinner. These had poor shelf life because we got too many splits. This was a larger leaf and more varied. This was a plant variety. I was trying to show that the variation in that particular variety was pretty high. This one was more uniform and less prone to cracking. This one was also uniform, but it had too many leaves that might crack. Again, something I never even thought of until I started running tests and went, huh, why are we having a problem here? And I'm going through hundreds of bags of stuff, looking at individual leaves one at a time. What the hell's going on? And I started to go, click. These very round spinach leaves are a problem. They split. And nobody knew that before until people started doing high volumes of baby leaf spinach in massive production quantities in salad dryers and selling them in bags at the store. That was a new thing people had to worry about as a plant breeder and as a grower and as a processor. Horrible photo. The original was much better, but these are varieties of strawberries that I actually worked on. These were some of the elite varieties that we grew when I started a strawberry breeding program a number of years ago. And just to show you different sizes and shapes and colors, again, you can't really see the level of differences the way I would want to because it's kind of a blown out, crappy copy of that photo. And this is why I wanted to do strawberry breeding. This is out near Seascape Village, south of Aptos, north of Watsonville. That whole region, we had hundreds of acres of beautiful land. And wouldn't you like to have that be your place of work all the time? That was some wonderful years. Anyway, that is it for the breeding.